Okay, Year 11, this is the B2 revision tutorial. We're going to focus on proteins, their functions and uses. So, some key phrases that you need to know. The first one is catalyst. Now, a catalyst is basically something that speeds up a chemical reaction, okay, without being changed itself. So it can continuously be used. Okay. Second one is an enzyme. Now, an enzyme is a biological catalyst, and some of the enzymes that you might need to know about might be lipases or even carbohydrases. And proteases. Now, if you notice, you can see that at the end, it's A's, okay? Anything with A's at the end is an enzyme. The active site, that's the part on the enzyme, so this bit here, okay, where this substrate will bind and there will be a chemical reaction. Finally, activation energy. Now, this is the minimum amount of energy that particles must have for them to be able to collide and react together. Curve, we can see that to be able, for the reactants to be able to react together and produce products, they need activation energy. Now, what you'll notice is with the introduction of an enzyme or a catalyst, what happens is rather than having a high activation energy, so lots of activation energy, what the enzyme does is it lowers the activation energy. So we need less energy in the system for the reactants to react together and produce a reaction. So one of the things that you have to understand is how enzymes work. So if we have a quick look at this, we can see that the green molecule is the enzyme. Okay, so this is the enzyme. We also have the yellow molecule, which is the substrate. Okay. Now, if we have a look, the enzyme has a particular shape, okay? And the shape is complementary to that of the substrate. So they fit together a little bit like a lock and key, okay? So only particular enzymes can break down particular substrates, and this is because of their shape. It has to be complementary. Now, what happens is when an enzyme and substrate collide, so they hit each other, they form a lock and key. Now, this lock and key can be called an enzyme substrate complex. Now, whilst they're together in this enzyme substrate complex, what happens is the enzyme is weakening the bonds or helping bonds to form to basically break down the substrate. Okay, in the case of digestion, we'd be breaking down maybe starch into glucose or protein into amino acids. Okay. Now, there are a number of different factors that affect the rate, okay, of enzyme catalyzed reactions. Okay, so that basically means there's factors that affect how quickly the reaction proceeds. So we have temperature, pH, the concentration of enzyme, the concentration of substrate, the surface area and the pressure. Now, enzymes tend to work best at a particular temperature and a particular pH. This is their optimum temperature, okay? This is where they might break down the most amount of product in a given time. Now, enzymes that work in the body are going to work well at 30 degrees C, okay, because that is body temperature. Now, depending on whereabouts the enzymes work or where they're catalysing reactions will depend on the pH. So, for example, an enzyme that works in the stomach is probably going to have an optimum pH of maybe 1 to 2. Okay, so it's a little bit more in more detail. Basically, outside of their optimum, in extremes of temperature or extremes of pH, what can happen is the shape of the enzyme can be irreversibly changed. Okay, so remember, the part of the enzyme that's really, really important is this bit. It's the active site. Should be okay. Let me just do that again. Okay, so it's the active site. Okay, now if in extremes of temperature and extremes of pH, what happens to this active site is it changes shape. So that means that it's no longer going to be complementary to the substrate. Okay, so the substrate isn't going to be able to fit in. Okay, so the substrate's not going to be able to fit in, it's not going to be able to form a lock and key, so the enzyme is not going to be able to break down this substrate. Okay, in a particular graph you can see that as we increase the temperature, we increase the amount of kinetic energy. So what's happening is the enzyme and the substrate are moving around more. If they're moving around more, it's more likely that they are going to collide, okay? And therefore you're going to get maximum rate of reaction, so you're going to get more product formed. Once you get to their optimum, so on this graph it looks like it's about 35, okay? This is the best temperature for the enzyme to be catalysing that reaction. But once we get past it, to higher temperatures, what we start to see is the temperature is irreversibly changing this active site. Okay, so it's denaturing it. You cannot say die. The enzyme does not die. It is denatured. It's changing shape. It's not living. Okay, so it doesn't die. So the enzyme changes shape, so the substrate can no longer fit, so it doesn't break down the substance. Now, if we look at pH, we can see that different enzymes might have slightly different optimums. So if we look at the black line, we can see this is a pH curve, okay, for pepsin. Now, pepsin is a protease that is in the stomach. Okay, so if you have a look, the optimum pH, the best pH for this protease, pepsin, to work at is about pH 2. If you go outside the range, so between 3 and 4, the enzyme's going to denature and it's not going to work. And if you have a too low pH, it's also going to denature. So either side of this curve, it's going to denature. The same with a different enzyme. So we can see this is trypsin, which is another protease. But this time, this one's optimum is about pH 8. And again, if you put it in a more acidic pH, it's going to denature the enzyme. If you put it in a more alkali pH, it's also going to denature the enzyme. So the reaction. Now, enzymes are produced in the mouth, the stomach, and the small intestine. Okay? And most of the enzymes are secreted by the pancreas, but they work in these areas. Okay? So enzymes essentially break down large food molecules into smaller ones so they can be absorbed through the wall of the small intestine and into the bloodstream. And then the blood can carry around all those digested products around the blood and into the cells where they're needed. Now, this is chemical digestion. Now, there are a number of different enzymes that you need to know about, okay? And after each of these slides, you might want to pause the slide and see what you can remember. But essentially, amylase is a type of carbohydrate. Okay. It's made in the salivary glands, pancreas, and small intestine. Okay, so that's where it's made. And what it does is it breaks down starch into glucose. So you can see it breaks this large insoluble molecule down into this small soluble molecule so it can pass through the small intestine and into the bloodstream. So although it is made in these three places, it works in the mouth and it works in the small intestine. Okay, so that's where it breaks things down. So the pancreas, for example, would make the enzyme, the small intestine would make the enzyme, the pancreas would release the enzyme into the small intestine, and it's in the small intestine where the reaction takes place. Or the salivary glands make the enzyme, saliva is secreted into the mouth, and it's in the mouth where the reaction happens. If we look at some others, so protease, look at this word, protease. It breaks down protein into amino acids. Again, the protease is made in the stomach, the pancreas, and the small intestine. Okay, so you can see the only difference here is that it's not made in the mouth, but it is in the stomach. It works, so it breaks down proteins in the stomach and the small intestine. So when in doubt in an exam question, small intestine is probably your best bet as to where something is made or works. Okay, again, lipase is made in the pancreas and the small intestine. Lipase breaks down lipids, which are fats. Okay, so lipids are broken down into fatty acids, and you can't quite see the words, so I'll write it here, and glycerol. So again, you can see you've got large insoluble molecules being digested by the enzyme lipase into small soluble molecules, which are fatty acids and glycerol. And again, this is working in the small intestine. Okay, so digestion that happens in the stomach. Now, as part of one of the process of chemical digestion, and to help with the enzymes in the stomach, so the proteases, remember they are made in the stomach and work in the stomach. Because proteases work at a low pH, so they work at about pH 2, the stomach secretes hydrochloric acid, which creates the perfect pH environment for the stomach enzymes to work and break down protein into amino acid. Now, the enzymes in the pancreas and the intestine work best in alkaline conditions. So if you imagine our food's moved down our esophagus, it's gone into our stomach, and it started the digestion process of proteins. As it moves into the small intestine, okay, the pH is still going to be quite acidic, so it's going to be 1 to 2. Now, enzymes are going to be secreted from the pancreas into the small intestine. So here's our pancreas, and it's going to be adding enzymes into the small intestine. Now, to prevent these enzymes from denaturing, the liver produces bile, and it releases that bile into the small intestine. Okay, now what that bile does is it neutralises the acid to prevent the enzymes that are being released from the pancreas from being denatured, because these enzymes work at a slightly more alkaline condition. So if we have a look at the role of bile, so the first function is to neutralise the acid. And the second function is a process called emulsification. So if we look at a fat droplet and we add bile, bile is a little bit like a detergent, like washing up liquid. And what it does is it splits up the large fat droplet into several smaller droplets. Now what that means is there's a larger surface area for the lipases to work on the lipid to break it down into fatty acids and glycerol. Okay, which means the rate of digestion will increase, so more fatty acids and glycerol can be absorbed into the bloodstream via or through the small intestine. So the last bit is what enzymes can do for us. Well, we use them in industry, in confectionery or sweets, baby food and biological detergent or washing powder. So the function of enzymes for sweets basically changes glucose into fructose using isomerase. So isomerase is the enzyme that breaks down glucose into fructose. Now fructose is basically sweeter, okay, so less of the sugar would be needed, so we can use fructose in slimming products that are going to help people gain less or not gain weight or gain less weight. Baby food, because babies' digestive systems might not be fully developed, we need to add proteases and lipases to start to break down the food. So it's much more runnier consistency, so it's easier for the baby to digest. And finally, biological detergent contains proteases and lipases, and what that allows to do is break down stains that you might have on your clothes. Downside to some biological detergents is some people have allergic reactions to the enzymes, and obviously we need to use lower temperatures so that the enzymes aren't denatured. However, that could be a good thing for the environment in terms of the use of electricity and energy, and therefore contribution to global warming. So I want you to have a go at this question. So it's a 6-mark question. Okay, and we're going to use something called busy. So what we're going to do is we're going to box the command word, which is describe. Okay, so it says describe how different parts of the human digestive system help to break down molecules of fat so they can be absorbed into the body, make reference to any enzymes involved, the products of digestion, and chemicals involved in the process. So we've boxed, we now need to underline keywords. So box the command, we're then going to underline keywords. And then scribble the extra vocab to get started. So let's think about this. So we've got digestive system, we've got fat, okay, we've got absorbed, we've got enzymes, we want the products of digestion and chemicals involved. So if we think about fat, we need to think about glycerol and fatty acids. We want to think about the role of bile and the liver. We might want the word emulsify, neutralise, we might want to think about things like mechanical digestion. Because it's talking about absorbed, you might want to go with small intestine, because that's where it's going to happen. Okay, you can keep adding your keywords, but that's going to give you a starting point. So pause the video. I'd like you to have a go at the 6-mark question, and then there is a mark scheme at the end where you can see how many points you've got. I hope the tutorial was helpful. You can obviously rewind it again to go over anything you're not sure of.